Now let's get into the last bit of how we can overly complicate genetics by not only looking at the genes themselves, but also look at how the environment can influence those genes. So we're going to start with our example of Siamese cats and Himalayan rabbits. Siamese cats and Himalayan rabbits are some animals that will have darker pigmentation around their nose, ears, paws, and tails sometimes, while the rest of their body remains more of a cream to a white. And the reason that they have this coloration is because of the environment, and we'll talk a little bit more about that in just a second. But some things we want to note before getting deeper into our example is that DNA is not an exact blueprint for life. It does serve as our instruction manual. It has all the recipes we need. However, we also can change how often we use each recipe. So some things that influence gene expression, meaning how often we use those genes. Do we use them very often? Do we use them rarely? Are things like diet and nutrition, exercise, stress, education, and climate. Let's look at diet and nutrition, for example. In the previous video, we talked about the over 180 genes that help contribute to height. However, diet and nutrition also plays into that as well. If you have a genetic makeup that tells you that you will be very tall, but you don't receive proper nutrition as a child when you're growing, you won't reach your maximum potential in height. Your growth will be stunted. And the reason being is that even though your genes dictate that you will be 6'6", if you don't have the proper nutrition, you don't have the building blocks to actually go through that growing process. And so your growth is stunted. Maybe you end up only 5'10" instead of that giant 6'6". Exercise is another one. We see that there is a lot of different diseases and conditions that are related to lack of exercise. Same thing with stress, when you're overly stressed and while you are students, and it's pretty much unavoidable, how you manage your stress can be extremely important in managing good health. For education, having a better education, understanding these things can lead to better decision making in what you eat and how you live your life. And with climate, especially things like pollution, exposure to certain pollutants does increase risks of things like certain cancers and can influence the gene expression in other genes. And the study of how these genes can be basically moderated or regulated and how they're expressed is called epigenetics. So epigenetics is a new study, a subset of molecular biology looking at how genes are regulated. We've talked about the basics of gene expression, but this is above and beyond that. This is how often we turn on or turn off genes. When do we turn them on? When do we turn them off? Do we ramp up production? Do we ramp down production? And this is something that's still being studied today. It's not something that we really know a ton about. So we're not going to really go into epigenetics beyond this. But let's go ahead and talk about these cats and rabbits. So we look at these cats and rabbits and we see that there's pigmentation at the ears, at the nose, and at the paws. The reason is that the dark pigmentation that gives the darker fur will only function under cold conditions. So the production is only going to happen in the regions of the body that are generally speaking cooler in temperature. The ears, the paws, and the nose are typically far enough away from the core body that they are cooler than the main body of the animal. So we see that the dark pigmentation happens anywhere that the body is slightly cooler, and wherever it's very much warmer, like the head and the central core, is generally speaking cream to white in color. Now, that being said, you could not pack ice onto these animals to temporarily create darker spots on them. That's not necessarily how that would work. First of all, you would have to wait for the hair to grow out in its entirety, and let's be honest, if you can get a cat to sit down and do that, you're basically a miracle worker. Now, some other things we want to look at is the dihybrid cross. Now, something that Mendel talked about was that all of these traits were passed independently of each other. Now, he did not have an understanding of genes and thought these were free-floating entities somewhere within the plant itself that he was studying. And he was right in that they were unrelated, but his reasoning was not quite correct. Granted, we have to give him the benefit of the doubt in that he had no way of knowing what we do now. He did not know about genes or DNA or chromosomes, anything of that nature. So even without all that and being slightly off on that, it's pretty amazing that he understood what he did, and that's why we call him the father of genetics. But let's go ahead and look at two traits and test this idea that these traits are passed independently by using a dihybrid cross. A dihybrid cross crosses two individuals where instead of following one trait, which we've been doing, we're following two. And so we see that we follow Mendel's law of independent assortment. Basically, that one trait does not influence the inheritance of another trait. So let's look at these cats, because I'm very happy that Pearson likes cat for their examples. But let's go ahead and look at these two cats, the mother and the father, both of which are heterozygous with short hair and white fur. The hair will be shown with L, and the fur color will be shown with W's. And so if we look at both the parents, since they have the same genetic makeup, we see that the gametes they can produce are dominant and dominant, dominant and recessive, recessive and dominant, or both recessive. So we notice that each of the gametes have one L and one W each. And basically these four different gametes are the four possible combinations of capital and lowercase L's and capital and lowercase w's that we have within this mother's genotype. When we fill in our Punnett square, we see we go from 4 squares to 16. And we have individuals who are dominant-dominant, dominant-recessive, recessive-dominant, and recessive-recessive. Now, when we're looking at this dihybrid cross, we see that most of the individuals look like the parents. 9 of them, these guys here, have the same look of short hair and white fur. And we notice that there are various combinations of homozygous dominant and heterozygous for both traits. But because we have the potential of making gametes with recessive alleles, we see 3 individuals who are orange in color. They're recessive for fur color, and so their fur color ends up being different from the parents. We also see, since we have recessive L's, that we have the potential to be homozygous recessive for hair length. And so we see 3 individuals who are recessive and dominant, giving them white fur that is also long. And then lastly, 1 out of the 16 possibilities is recessive across the line, recessive for both fur length and fur color. And so this individual ends up not only orange, but also long-haired. And so we get a classic 9:3 to 3:1 phenotypic ratio. Now, we still basically see a 3:1 ratio for each trait when crossing heterozygotes, but we're crossing them concurrently. Since it's independent assortment, we're saying that these traits are inherited independently from one another. But when we're looking at real genetics, we see that this is not always the case for every single gene and every trait. There are some characteristics that defy Mendel's second law of independent assortment. For example, most people with red hair also have fair skin and freckles. We would call these traits linked genes. And linked genes are not necessarily linked by some special sort of circumstance or material or molecule. All it is that they're really close to each other on a chromosome, and so it's unlikely that they're going to be separated over from each other during the process of crossing over. So let's go ahead and look at that with these linked genes. So here are the homologous chromosomes. We see the maternal copy and the paternal copy. Now, we have 3 different genes we're looking at. One that's like in shades of blue, one in shades of yellow, and one in shades of red. The ones that are close together are considered linked genes, whereas the ones that are farther apart are considered non-linked. Again, they're considered linked because they're just so physically close in proximity. So when we go through crossing over, the likelihood is that these 2 genes will go together just because they're so close together. whereas the non-linked genes are far enough apart that they'll probably cross over and not necessarily stay together. And so when we see at the very end, we see we've crossed over some bits, and we have a new combination with this light blue, yellow, and red, whereas this one now has dark blue, light yellow, and pink. So we see that when alleles are really close together, that crossing over process is less likely to separate those alleles. So that's why certain traits seem to go together. It doesn't mean it's impossible for someone to have red hair and darker skin or not have freckles. It's just it's more likely to happen simply because they happen to be in close proximity. But it's not impossible to separate them using crossing over. So we've really explored a lot in heredity. We started with simple dominance, looking at a simple dominant and recessive relationship between two alleles, giving us only two different phenotypes. We performed test crosses to determine the genotypes of unknown dominant phenotypes. We've looked at sex-linked genes that are related to being male or female. We've also explored incomplete dominance, where you don't have a typical simple dominant recessive relationship and instead see a blending, as well as codominance, where instead of a blend, the heterozygote has two alleles expressed discretely and equally, and that for many different things, many different traits, a population can have many different alleles, although an individual will only have two. We looked at how a single gene may influence many traits with the concept of pleiotropy, and that many genes usually influence one single trait, known as a polygenic trait, and that in many cases genes can be linked together just because they are closely related in terms of location on a chromosome. So there's a lot that we covered in this unit, in this chapter. Definitely take the time to understand the tools and the concepts from this unit. It's a lot to digest, but once you get the ball rolling, it's not so bad. And it opens a lot of doors and opens your eyes to understanding how you are the way you are and why you look, act, and behave the way you do.